Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the water use efficiency. So, friends, this is also a very important topic, and in this lecture, we will discuss the different aspects of the water use efficiency. Okay. First, we will discuss that what is what is water use efficiency. Then we will discuss the some formulas that how we can calculate the water use efficiency. Then we will discuss about the relationship of the photosynthesis and transpiration in regards to water use efficiency. Then we will discuss about the water use efficiency of C3, C4 and chem plants. Then we will also discuss the uh, measurement or methods to measure the water use efficiency. Then we will also discuss the factor or plant traits, plant traits which affect the water use efficiency. So friends, you know that in today's agriculture that climate change and abiotic stresses are the major problem which reducing the yield and productivity of major agriculture crops. Okay, so also the drought stress and scarcity of water is a global problem, and it's affecting the global food security. Okay, therefore it is crucial to increase the water use efficiency. So the main motto of the increasing water use efficiency is the more crop per. Okay, so this is the main motto of the increasing the water use efficiency. Okay, so we will start that what is water use efficiency. So as the name shows, water use efficiency. Okay, so as name show, so in simple term we can say this is the capacity of plants to use water. Okay, so this is the efficiency by which plant can use the available water. Okay, so what is the available water with the plant and how can efficiently it utilize for the metabolic activities. That is known as water use efficiency. So if we will define the water use efficiency, so the formula is the mass of CO2 fixed, okay, upon the mass of the H2O transpired. Okay, so we can define the water use efficiency in mass basis by the mass of CO2 fixed, means how much CO2 fixed by the plant and how much H2O or water transpired by the stomata. So this is the mass basis. Also we can define on the mole basis. So mole of CO2 fixed upon the mole of H2O transpired. Okay, so we can define the water use efficiency in the mole basis. Okay, also some agronomists defined as a water use efficiency as by upon T. Okay, by means this is the yield and ET means evapotranspiration. Okay, so this ET includes the evaporation from soil surface, okay, then transpiration, okay. So evapotranspiration means evaporation plus transpiration. Okay, so we can define the water use efficiency in different terms in the photosynthesis upon transpiration over water use efficiency. Okay, also we can define that how much biomass produced upon how much water applied in the irrigation form. So we can define water use efficiency in different terms. Okay, so you know that to understand this water use efficiency, we have to understand the water uptake and transport. Okay, second, we have to understand the transpiration process. Okay, so how we can understand this water efficiency? By water uptake and transport and transpiration. So you know that this is the soil surface and water is present in the soil surface in solution form. Okay, and this is the plant, this is plant root, this is stem, and these are the leaf. Okay. So in the soil, water moves by the mass flow, by the diffusion, and you know that water always move from higher concentration to lower concentration. So soil surface have high water content and this leaf surface have less water concentration or less water potential. Okay, so water is uptaken by the root. Okay, then water move in, this is in the roots tissues and reach to the xylem. Then water flow in the xylem and reach to the leaf surface. This is known as long distance water transport, and in the root it's known as short distance water transport. Okay, then in the leaf surface, in the epidermis, there will be a pore-like structure. Tiny pores are present, which is known as stomata. Okay, so this stomata are important for the facilitated diffusion of CO2 and H2O. So this stomata is crucial for the CO2 fixation or transpiration. Okay, so they are the pores for the entering the CO2 and exit of the or transpiration of the H2O2. So the stomatal movement is crucial for the both photosynthesis and transpiration. Okay. Also, we can say that photosynthesis is the benefit for the plant, and transpiration could be the cost of the plant. Okay, so this is about the stomata role in the water use efficiency or water maintaining of water potential. So friends, you know that transpiration and photosynthesis. Okay, so transpiration is generally not a problem because this is important process which reduce the heat load on the plant. You know that plants are sessile in nature. Okay, they cannot move from one place to another place. So during daytimes there will be a high temperature. Okay, so this increase the canopy temperature. If there is a no transpiration process, then it may be possible then in some minutes the plant canopy temperature reach up to 100 degrees centigrade, which can kill the plant. So transpiration is not a problem, but due to transpiration, due to excess transpiration, or in the water scarcity areas, reduction in the reduction in the cell or leaf turgidity or water potential of the leaf is the major problem. Okay, because it can affect the metabolic process. Okay, so you know that about 97% water uptake by the root is 95 to 97, more than 95 you can say, transpired through the plant. Only 2 to 5% is water is utilized for the metabolic reactions. So reduction in the water potential or turgidity is the major problem. So you can say that stomata closing can improve the water use efficiency, but it also reduce the photosynthesis or CO2 fixation or CO2 diffusion. Okay, that's why in some case or rainfed agriculture we use the anti-transparent. So already in previous lectures we have discussed that there are different types of anti-transparent which partially close the stomata to increase the water use efficiency and by only some affecting the photosynthetic rate. Okay, other than this, plants, there are three types of plant which are the C3, C4 and chem plant. Okay, so you know that chem plant have photoactive stomata. Chem plant example are like agave, okay, opuntia, okay. So these plants have photoactive stomata which are opens during night time, okay, and during the night time there will be a high atmospheric humidity, so BPD between the leaf and air is less, that is vapor pressure deficit, so there will be a less transpiration. Okay, so they have highest water use efficiency. Okay, then there are these two are the important group of plants and majorly plant on this earth are C3 types. The example is rice, wheat, and in the C4 group it include the sugarcane, maize and grasses. Okay, so these two plants also have different photosynthetic reactions. Like in C3, Rubisco is the major CO2 fixation enzymes, but in this C4 plant, Papcage and Rubisco are the main or important photosynthetic enzymes. And also in C4 plant, there is carbon concentration mechanism. Also in C4 plant, there is no photo respiration. So the C4 plant efficiently use the CO2, or they have higher CO2 fixation efficiency. Okay. Also they have less stomatal conductance. So they have high water efficiency. But in case of C3, they have less water use efficient plant. Okay. So if you will see that the water use efficiency for C3 plant, it's near to 1 to 3 gram of CO2 by the per kilogram of H2. Okay. For C4, the value will be the 2 to 5 gram of CO2 per kilogram of H2O. And for CAM plant, this value vary with 10 to 40 gram of CO2 per kilogram of H2O. So what it mean this? This mean that C3 plant fix only 1 to 3 gram of CO2 by the transpiration of per kilogram or per liter of H2O. Okay. And C4 have high efficiency. They are 2 to 5 gram of CO2 per kilogram of H2O. And CAM plant have highest water efficiency. It is vary with 10 to 40 gram of CO2 fixed by per kilogram of H2O. Also, the water use efficiency is inversely proportional to the transpiration ratio. Okay. So transpiration ratio is defined as the mass of water transpired upon the dry matter produced. Okay. So generally this for C3 plant, the transpiration ratio is 600. For C4 it is 300, and for CAM plant it is 150. Means C3 plant have high transpiration ratio and less water use efficient, because this is inversely proportional. Okay. So this is about the stomatal regulations and water efficiency. Now there is a one scientist that is known as Peschiora model. Okay. So they propose a model in which the formula is Y barabar water use efficiency into water use into harvesting index. It is also second way we can say T T into TE into HI. Okay. So yield, Y बराबर yield is depend upon the water use efficiency. Water use means how much water is used and how efficiently this water used, and the harvesting index, like that transpiration and transpiration efficiency into harvesting index. So this is about the, also you know that C4 plants are more advanced types of plant and they can grow well under the highlight intensity, highlight intensity, water stress, water stress, and challenging environment. Okay, so they are evolved for the high water use efficiency. Now come to the measuring the water use efficiency. Okay, so there are several methods to measure the water use efficiency. First one is the gravimetric method. Okay, so this is the traditional method of the measuring water efficiency. So in this we can measure the water use efficiency by weighing the plants in the pot and daily adding of required water. So what we will do in the gravimetric method means, if we conduct a experiment in the pot experiment, then we have to measure the plant width or pot width daily, and accordingly we will add the water. Okay, and also currently lysimeters are used for the measurement of water requirement. Okay, and third is the ERGA, that is known as infrared gas analyzer. Okay, so this instrument measure the photosynthetic rate upon the transpiration. So by measuring the photosynthetic rate and transpiration, we can measure the water use efficiency. Fourth method is the by the carbon 13 isotopes measurement. Okay, that is also known as carbon isotopic discrimination. Carbon isotopic discrimination, and the instrument that is known as IRMS, isotopic ratio mass spectrometry. So isotopic ratio mass spectrophotometer is used for the measurements of carbon isotopes, and you know that carbon and is diffused by the stomata and simultaneously diffuse the H2O. So the carbon concentration is determined the water Eugyptian. So also in the second lecture or previous lecture, we already discussed about these slides, that how that in the nature there is two types of carbon is present, that is C12 and C13. Okay, so this is the isotopes of carbon. Okay, so this is present in very high amount in the nature. Nearly 98.9% of carbon in the form of CO2 in the form of C12, and only 1.1% is available in the form of C13. So this C13 have heavy molecular weight. Okay, so this C13 have heavy molecular weight. Now this is known as isotopic effect. Okay, so one is the isotopic effect, that these are present in very less concentrations. Also leaves discriminate against the C13, because that there will be a this C13 है high molecular weight, so their diffusion also is very low, so it reduces the ratio of the C13. Also this IRMS, isotopic ratio mass spectrometer, calculate by the formula R samples upon R standard minus 1 into 100. So R standard for the C13 is the minus 8 per mil. What is per mil? This is the per thousand. Okay, and it is measured at the standard taken from the Vienna PD Belmonite PPDB. Okay, so these are the discrimination factor for delta 13C. This is minus 28 and minus 14 for the C3 and C4 plant, because in C3 the main enzyme is the Rubisco, and C4 it is package. Okay, then C12 and C13 diffusion fraction is the 4.4 per mil due to their molecular weight. So when you will close the stomata, or there will be a less stomatal conductance, then the ratio of the C13 increased, and it shows the higher water efficient. That's why in C4 plants have more delta 13C. Okay, so this is about, and this is the method of long term measurement of water Eugyptian. Also these carbon isotopes can be used for the long term climate change. Okay, also the measurement on the O18 isotopes used for the evapotranspiration measurements. Okay, so these are the some methods to measure the water use efficiency. Now come to that what are the parameters which affect the water use efficiency. So this is the plant. Okay, this is leaf cell, this is stem, leaves, and this is root. Okay, so the radiation interception, okay, on the leaf surface and the soil surface increase the evapotranspiration. Okay, then heat load. Okay, heat load, or you can say the air temperature, that is also affect the water use efficiency. Then leaf morphology, like stomatal conductance. Okay, so it also affect the water use efficiency, already discussed. Then also the water uptake and transport, means how efficiently plant can uptake the water and transport to the leaf surface. Also the resistance. Friends, you know though that there are several types, several resistance, like boundary, leaf boundary, layer resistance, then different cuticular resistance, also stomatal resistance. They also regulate the flow of the water and CO2. Okay, so they also affect the water use efficiency. Then root system architecture, that is root system architecture. So this is also affect the uptake, transport and efficiency of water utilization. So friends, this is about the water use efficiency. Hope you understand that what is water efficiency and what are the formulas and how the stomata regulate the water use efficiency, and how this we can measure the water use efficiency and different methods. So friends, thank you very much for today's lecture. Hope you understand that what is water use efficiency, the concept water use efficiency. Thank you very much.